As scientists, we make 1,000 small decisions to make a robust method. Every decision that we are making is with the end goal in mind, and that's the patient and saving lives.
Welcome to Data and Biotech, a podcast from Cordi where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Callie, Celichowski, welcome to the Data and Biotech podcast.
Thanks, Ross.
Awesome. Well, we're here at the AAPS PharmaSci 360 conference and excited to talk with you about your work and about Veloxity Labs. Can you just give us an introduction to you and your background and the work that you do?
Yeah, sure. So Callie Celichowski, and my background is all business related. I started on a marketing team at a really large CRO where they did great training and kind of introduced me to drug development, and then moved to smaller, mid-size and small CROs, more on the sales side.
Yeah, awesome. And so would you introduce us to Veloxity Labs?
Yeah, so Veloxity is a bioanalytical mass spec-based CRO. We are located in Peoria, Illinois, and really focused on very early screening, drug discovery, all the way through IND enabling and clinical bioanalytical support. So really partnering with clients to do everything from start to finish, mass spec based.
As we were preparing for this podcast, I know that one of the catchphrases or taglines for Veloxity Labs is this idea of speed with purpose. Mm-hmm. Can you sort of unpack what that means in the context of how you work with clients?
Yeah, I think there are a lot of CROs that can provide a set of data, and they can send it over and they can give you an Excel, they can give you a Word report that has, what was found. But when you have questions or you want to really discuss and get into the nitty gritty of what that data means, I think that Veloxity really is always there for you. We're doing not only the delivery very, very quickly, but if you have questions, you call, you text, you ask the questions, and our people are going to be there start to finish to give you what you need.
From the client perspective, or from the sponsor perspective, what do you think they're looking for in a CRO? How do they evaluate which CRO they're going to select, and why do you think they end up with Veloxity Labs?
I think that a lot of our clients have a relationship with one of us, from the past, and we've gotten to know one another. We don't, run before we walk. We kind of start with a smaller project and then kind of dive into more deeper, longer-term strategic partnerships. I think that really clients are looking for a partner that they have kind of a one person to go to, a scientist. So our scientists work on things from the start to finish. We don't do a handoff from method development to method validation to sample analysis to recording. You have API that is working with you on every step of that process, and they are your main contact versus, we're going to start with the development and then somebody's going to do the validation, and then yet somebody else is going to do the sample analysis. And that works well for, I think, larger institutions that have more of a machine type of philosophy, whereas we are really focused on that relationship and getting you, from start to finish.
One of the things that interested me about Veloxity Labs is sort of your work with peptides and, and especially, the idea of working with GLP-1 receptor agonists, these drugs that are really becoming really transformational for weight loss and diabetes treatment and a variety of other indications that are out there. So I'm interested in, number one, what kind of work you all have done, or how you all are positioning yourself for the work that needs to be done with regard to these peptides.
Yeah, I think that we are. It's trending, so there's a lot of publicity and advertisements and all of that good stuff going on about different, GLP-1s and other, peptide type work that's in our wheelhouse. So trending, that's one thing. There's always the popularity, and there are things that are going to go in ebbs and flows. And so I think that this, is a big one right now. I think that they're trying to put their best foot forward in making sure that they're going about this in a, regulated fashion. And so I think that being able to get additional products to the market that maybe are more efficacious, but yet still safe, is really, really important. And so, the companies that are working on that are ideal for us.
Yeah. Is there anything that you see on the horizon for, how the CRO model as a whole, or how Veloxity Labs in particular needs to change or adapt as molecules become more complex, as the type of analysis that you're doing or the nature of the assays that you need to apply needs to change in order to?
Yeah, I mean, I think we always, as an industry, have to be adaptable. I think gene therapy, for example, that was all we were talking about maybe a year and a half, 2 years ago, and that particular segment of our industry has had a rough go of it lately. And so it doesn't mean that it's forgotten. It doesn't mean that it needs to go away. We still need to find solutions, but we also have to be ready for what's next. And I feel like the GLP-1s and different, peptide, more complex analysis is necessary. And I think that Veloxity has done a really nice job not going overboard, but staying, kind of on top of some of the trends.
As you look forward to 2026, is there anything, in terms of how Veloxity Labs' footprint or machinery or the way that you operate might change that you're particularly excited about?
I think that, just continually growing is important, but growing with purpose, not growing just to grow. We don't want to be the biggest CRO. We don't want to be a one-stop shop for everyone. We want to be experts in bioanalysis from early screening all the way through whatever your endpoint is as a client.
Well, Callie, thank you so much for joining the podcast today. Isa Kupke, welcome to the Data and Biotech podcast.
Thank you so much for having me.
Awesome. Well, just to kick us off, would you give us an introduction to your background and your work at Veloxity?
So I'm Isa Kupke. I'm originally from South Africa. That's why I have the accent. I started in natural product research, and I through that, I got into mass spectrometry, LCM, SMS. So I came to the US. I did some work on American plants and their traditional uses. And then I came to Veloxity, and I've been loving it. So I'm working as a scientist now at Veloxity, helping our PIs with doing work for our clients and helping them make the decisions that they need to do.
Yeah, awesome. So, working as a scientist, a CRO, I could imagine a lot of things about the different types of problems you get exposed to, but would love to hear from you about, do you have any example projects that you're willing to share, or, give us an overview of what your work looks like?
So we work mostly currently on small molecules, but we are moving more into peptide work. And peptides are notorious to be a little bit more difficult to work with. They need a lot of preparation during method development, tuning. They have multiple charge states that affects the sensitivity. And we try our best and get the best robust method that we can for our clients, so that even if they are in the preclinical phases where we're dealing with just making decisions going into GLP work, and regulated work, we're really trying to get those methods as robust as possible, really understanding the stability of an analyte, of a peptide, and so that once we go into the actual regulated work, that we don't have any surprises, and that the data we are getting is as robust as possible, and that it is ready to be submitted to the FDA.
Yeah, so I mean, I think for people who aren't familiar with the work of CROs, like connecting the dots between a drug that you might get in the pharmacy to the kinds of work that you're doing in the lab in order to like characterize these preliminary drug candidates to prepare them for bringing them to the market. Like it's hard to understand how these things connect to each other. But my understanding is that, what the kind of work that you're doing with mass spec is to try to like interrogate the nature of these preliminary drugs and like quantify some of the aspects of it, such as stability that you just mentioned, so that you can help your clients to answer the most critical questions that they need to answer as they're preparing to launch them. So would be interested in hearing from you, like what are some of the most important questions that your customers are asking about these preliminary drug candidates? And then like, how do you attempt to answer those questions with the kinds of mass spec methods that you bring to bear?
So a lot of the drugs that our clients do, and they are mostly in the preclinical phase. So they want to know, does their samples, do they have a toxicity at any certain level? What are we detecting in the samples that they are sending during different dosing stages? So a lot of toxicity type of toxicology type of work that we are analyzing the drugs in. I think also efficacy, and our clients also want these results and this data fast, and that's what Veloxity is all about. I think it's so important for them to have a CRO partner that can provide data fast to them so that they can get the drugs to the patients as quickly as possible.
Yeah, that makes a lot of sense. And so, my understanding is that you're attempting to understand, interrogate the toxicity or the efficacy, but then you're also sort of developing methods for the detection of these properties. Am I thinking about that?
Yes.
Okay.
Not in that sense. So what we do is we want to quantify the levels of drugs or the level of the drug, either in certain matrices that the client might send to us. We get samples, we get blood samples, whole blood samples, plasma samples, tissue samples, and our extraction techniques vary based on the type of analyte, just so that we make sure that we don't get in any interferences once we get to mass spec, and looking at the concentrations of that specific analyte that they are interested in that matrix that they are sending to us. And that is what the mass spec is helping us with.
So my understanding is that you're using mass spec to like characterize all of these different quantifiable traits of the drug candidate that's coming in, and then you're providing that data back to the client. Are you providing any sort of like narrative support or guidance with regard to the data that you're providing back?
We mostly try to provide data that is easily interpretable for our clients. If they have more questions surrounding data and data trends, they can definitely approach us. But direct discussing the data or making very important decisions, such as for the efficacy, that is not what we currently do. But we try to make sure that the data that they are receiving is interpretable and that they can use it moving forward with their regulated studies.
Yeah, that makes a lot of sense. And obviously documentation of like the methods that you're applying in order to generate the data, I'm assuming as well, right?
Yes. So when we even with non-GLP work, all our methods are documented. Every step, every decision that we are making, we document that so that it can be moved from one phase or one test or where the client currently is. It can move through, and if there's maybe a scientist that's not there that day, and a scientist needs to take over, that the work never stops and they can just continue with that.
In our previous conversation with Callie, we were talking about, we discussed, this idea of peptides and GLP-1s. And, what I've heard is that the nature of peptides makes, and you actually shared this a little bit earlier, that peptides are harder to understand or harder to interrogate using the tools that you have available. Would you mind just unpacking a little bit about what makes it harder? And maybe that can lead into, why there is this need for the 8600 for, understanding, mass at a higher degree of accuracy.
Yes. So currently, peptides are super sticky, so they just stick to everything. And we definitely adapt our methods to account for that. Peptides are also very large. With the current 6500, the mass is too large, or the peptide is too large to really be effectively analyzed by the mass spec, and we have to do some digestion. That digestion normally takes a day. It also comes down to when you're building the method, the analyte or the peptide has multiple charges, and that affects the sensitivity that we are seeing. So it's difficult to go to really low LOQ levels where our clients might need that in deciding what levels the drug is effective at or toxic at. And so what's really great about the 8600 is that we can just pop that peptide right in there, and we can get data that we could have never seen in the 6500. We can go to concentrations that we couldn't have before with the 6500. The 6500 is still great for small molecules, but saving time with just the method development, the tuning, because peptides require on-column tuning, and it's a lot more difficult than just a small molecule putting it on the instrument, on the 6500, tuning it, and you have a method or a pretty decent method in like 10 minutes. Peptides takes much longer. So the 8600 is definitely going to save us a lot of time in developing a method when we are extracting the peptide digestion, protein digestion. It's going to be really fun. I'm so excited to get hands-on experience with it.
Yeah. So, first, I've never been exposed to the 8600 system before, but my understanding is that it's called HRAM, or high resolution accurate mass. And so the idea here is you can sort of measure the mass at a much higher resolution to many more decimal places of accuracy in order to detect something like a GLP-1 that might be in a sample, so you can then sample for much longer as the peptides are sort of diminishing in concentration, and that that sort of that the idea here is that sort of helps to characterize the way that the how much remains in the body over the long term, having the drug in your system. Am I understanding that correctly, or how would you update?
So sometimes when we do testing for a client, their dosing would go for. We normally have a pre-dose sample, and then once they start dosing, it could be various time points where they are collecting samples. And normally the concentration goes a little bit more down as you reach longer time points. Having the 8600 being able to detect analytes even at those low concentrations really helps the clients make the better decisions and faster decisions for the drug and moving it forward, because all those decisions add up to time and money and a patient's life. So that's so important, is to get the most accurate data and the most data that you can to make the correct decision.
Yeah, I think you all mentioned that you're one of the only CROs in the United States that has access to one of these machines, is gaining access.
We are the first. And I think there's only 2 equipment, I think, currently that has been actually manufactured. And we are the first CRO in the US to have this piece of equipment. And we are so excited to test it and to run samples on it.
Is the reason for that it's so new, it's just been developed, or that it's relatively expensive, or like how are it, or that you all are so excited about the capabilities of it that you've just gained access?
Oh yeah. So having the capability to use it, I'm really excited about the Xeno part and learning more about that Xeno trap part of the mass spec. It's pretty expensive piece of equipment. So that's why not a lot of CROs currently have that, and there is some training that goes into that, but just being able to go down to such low LLOQ levels and it being so sensitive and saving that much time with peptide work and being able to do some protein work on it as well is really going to be fun, and I think expand what Veloxity can do, and really staying at the forefront of bioanalysis.
Yeah, I mean, one of the other benefits that, as I was sort of reading up about the 8600 was this idea that you could move into microsampling. So, not have to take such large blood or tissue samples from patients in order to be able to interrogate them. Am I understanding that correctly, or is that is that something that you see on the horizon with the availability of this machine?
So currently we do have the capabilities of collecting samples or receiving samples from a client that does micro sampling. So we do work with Tasso and the dry blood spots, and we are with the 6500 we can get pretty good data from that, but sometimes with more difficult analyte that is not as stable, getting really into those low LLOQ levels that goes into picomolar or things like that. That is really difficult with some analytes, and I think the 8600 is really going to give us so much more information that we couldn't have.
And we've mentioned LLOQ multiple times. It's a lower limit of quantitation, right? And that's sort of like a measure of the sensitivity of the instrument that you're using, if I'm understanding correctly.
Oh, yeah. So it's going to that lowest concentration that a client wants. So the LLOQ is the lowest concentration that a client wants to measure. And sometimes it's really difficult to hit that target, especially like with a little bit more of a sensitive type of analyte or a drug that's not as stable, or there could be a lot of interference from a matrix, especially that's also a problem with peptides. There's a lot of suppression and things like that, and it's just hard to then get to that lower levels of an analyte. And so I think the 8600 is going to be really great.
You've got clients who want to be able to detect very small concentrations of drug in a sample, and sort of like interference from the matrix is like this idea that there are like other chemical elements present in the sample that might like introduce noise that interferes with your ability to detect those very low concentrations. And if I'm understanding correctly, that one of the reasons why they might want to detect those very low concentrations is like, I don't know, understanding how quickly the body metabolizes the drug, or understanding how much is left over so that they can think about dosing. Exactly. Am I thinking about that right?
Exactly. Yep, that's correct. Yeah. So clients want to know at those lower level how the body is, like you say, metabolizing them, and at what levels are the drug present during a specific time, and that will just help them make the decisions for absorption, distribution. So they're looking at all that at me.
Is there anything that you feel like people don't understand about the work of bioanalysis that you think they should understand, or like anything unexpected that you've encountered as you've sort of grown up as a scientist in this field?
Yes. There's definitely, I feel like a misconception is that, or that people don't know, is that as scientists, we make 1,000 small decisions to make a robust method. Every decision that we are making is with the end goal in mind, and that's the patient and saving lives. And that is why speed is so important for Veloxity. Our philosophy is treating a disease one sample at a time. And we always keep the patient in mind. And that's what we know, why we know speed is so important, is to really help the patient and helping our clients make the decisions to save lives.
Are there any what are some examples of some of those small decisions that you that you make when you're going through your workflow?
Mm-hmm. So we are making decisions for robustness, for data that would enable our clients to make the fast decision, to make accurate decisions, to make decisions that can be transformed into a viable product that saves lives.
Thank you very much for joining. Where can listeners go to learn more about Veloxity Labs?
Yes, so they can go to our website. It is veloxitylabs.com, and they can reach out to either me or Callie, and we'll send them more information.
Great.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.